Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing an exam review for Biology 1408, Exam 4, our last exam for the semester. We're almost there. This exam will cover chapters 11, 12, and 17. So let's go ahead and get started with those important points that you need to know so that you're successful on the exam and that you can finish strong. So let's start with chapter 11, dealing with meiosis. Please know the difference between what a diploid and a haploid cell is. What else? You should know that sperm and egg are haploid. The sperm and egg are haploid. So remember, the sperm and egg are the gametes, gamete cells, and that they come together, the sperm and egg come together in a process known as fertilization to form a diploid zygote. The zygote, which means the fertilized egg, is diploid. Next, we want to really understand the process of meiosis. What I want you to know about meiosis is that the first division of meiosis, remember M1, the first meiosis division? Meiosis 1 is termed reduction division. And what was meiosis 2 called? Do you remember? I'm going to skip ahead. Meiosis 2 is called equational division. So please remember that for the exam. What else do we want to know? We want to know that meiosis involves two meiotic divisions. Remember, there's meiosis 1 and meiosis 2. And we need to know that before meiosis 1 and meiosis 2, there's a process known as interphase, right? There's interphase. And remember interphase from chapter 10, we have G1, S, and G2 subphases. After interphase, we have meiosis 1, and then meiosis 2. Now, I want you to realize something. There's interphase, then meiosis 1, then meiosis 2. What I want you to know for the exam is that there is no interphase between meiosis 1 and meiosis 2. So there is no interphase step between meiosis 1 and meiosis 2. We should know what a tetrad is. Okay, when do tetrads form? Do you remember when tetrads form? Here it is. During meiosis 1, remember meiosis 1 begins with prophase 1, tetrads form, and then what? Crossing over happens. So I want you to know that during prophase 1, tetrads form and crossing over happens. Great. And then what happened during metaphase 1? I want you to know that during metaphase 1, the tetrads line up. The tetrads line up at the center of the cell. And by what process do the tetrads line up at the center of the cell? Remember that? They line up by independent assortment. So we want to know that for the exam. We want to know that during metaphase 1, tetrads line up, and not only do they line up, They line up randomly by a process known as independent assortment. Next, we want to know what happens during anaphase 1. During anaphase 1, the tetrads break apart, right? So what you need to know is what's written here in bold. Non-identical sister chromatid pairs separate from homologous sister chromatid pairs. What that means is the red sisters separate from the blue sisters, or the maternal homologous pair. The maternal sister chromatid pair separates from the homologous paternal sister chromatid pair. Essentially, homologs separate. Homologs separate. Okay, in anaphase 1, the homologs separate. And then there's telophase 1. What happens during telophase 1? Remember, at telophase 1, you have two cells, and these are haploid, two haploid cells. Two haploid cells, and I wrote it here and I underlined two haploid cells form at telophase 1. Next, remember there is no interphase between meiosis 1 and meiosis 2. We're heading directly into meiosis 2, and it begins, with prophase 2. And here, during prophase 2, we need to know, that sister chromatids form. During metaphase 2, those sister chromatids line up. See them lining up. During anaphase 2, the sister chromatids separate from one another. And then you have telophase 2, where it ends up with 4 cells. Now, this is what I want you to know. This is a very important point. This is the final result, which means the whole point of meiosis. The final result is 4 genetically distinct cells. And these cells are haploid. And what are these cells called? These cells are called the gametes, aka sperm or egg. Awesome, that's what we want to know. We can skip this part. In fact, the rest of these slides we can skip. All right, so these are the major points we want to know about meiosis. I touched on all the important parts for the exam. Please know these parts for the exam, and I think you'll be just fine. Let's take a short break time with Gizmo and Wicket, and we'll be right back with Chapter 12. All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and continue with Chapter 12 of our Exam 4 review. Chapter 12 deals with patterns of inheritance and heredity. You should know the definition of heredity as it is stated here. We should also know about Gregor Mendel and his hybrid crosses. What do we need to know about the hybrid crosses? Let's skip to the hybrid cross. This is Mendel's famous mono-hybrid cross. And these are the things I'd like you to know. I'd like you to know that a monohybrid cross always begins with true breeder parent. See what it says here? True breeding white parent, true breeding purple parent. So it always starts with true breeders. Okay. Secondly, I want you to know that when the monohybrid cross is performed, we end up with a 3-to-1 ratio of dominant to recessive phenotypes. So we end up with 3 purple flowers for every white flower. Again, we have a 3-to-1 phenotypic ratio. So monohybrid crosses give us a 3-to-1 phenotypic ratio. And remember that this 3-to-1 phenotypic ratio is actually a 1-to-2-to-1 genotypic ratio. It's a 1-to-2-to-1 genotypic ratio. All right, what else do we need to know? We need to know these terms here, right? So know the terms, the term genes, allele, homozygous, heterozygous, genotype, phenotype, okay? And know that the monohybrid cross taught us this principle, okay? The monohybrid cross taught us the principle of segregation of alleles, that 2 alleles for a gene segregate during gamete formation and are rejoined at random, one from each parent during fertilization. So I want to know that the monohybrid cross taught us the principle of segregation of alleles. Great, that's what we really want to know. I'm not going to have a Punnett square on there for you to solve. However, I do want you to know this about the dihybrid cross. I want you to know in a dihybrid cross, this results in a 9-to-3-to-3-to-1 phenotypic ratio. We have a 9-to-3-to-3-to-1 phenotypic ratio. So see this? We had 9 round yellows, 3 round greens, 3 wrinkled yellows, and 1 wrinkled green. So it's a 9-3-3-1 phenotypic ratio. What else do I want you to know about the dihybrid cross? That is the principle of independent assortment. The principle of independent assortment. The dihybrid cross taught us the principle of independent assortment. And this means that alleles of each gene assort independently of one another. So for example, yellow and round assort independently of one another. Shape and color are 2 different alleles. They assort independently of one another. They're 2 different genes, 2 different genes. So the alleles of each gene assort independently of one another. Okay, great. And we're not going to deal with the test cross for this exam. So that's about all I want you to know for chapter 12. That should be pretty straightforward for you. Let's hop right into chapter 17 after a short, another break time with Gizmo and Wicket. Hey everyone, welcome back from break time with Gizmo and Wicket. Let's continue on with Exam 4 review for Biology 1408 with Chapter 17, Biotechnology and Genomics. And I'm going to highlight some of these important points to know. From this slide, I wanted you to realize that you can use what are called microarrays. This is a microarray. You can use microarrays to determine which genes are turned on or off in different tissues of organisms. Okay, so on this slide, I wanted you to know about this microarray, and you see wherever you see a red line, the gene that corresponds with that microarray location is turned off, And wherever you see a green line, the gene that corresponds with that location is turned on. And on slide 2, I wanted you to understand about antibiotics. And antibiotics were discovered, and they are indispensable for fighting bacteria. Antibiotics fight bacteria. From this slide, I wanted you to know that we have techniques to extract DNA. We can extract DNA. We can then visualize the DNA by running it on an agarose gel. We can amplify DNA. What was the technique for amplification of DNA? Remember, that's called PCR, or polymerase chain reaction. I want you to know that PCR is used to amplify a portion of DNA, a sequence of DNA. Next, I wanted you to know about southern blotting. Southern blotting is used to identify particular sequences in DNA. Southern blotting is used to identify particular sequences of DNA. Next, we talked about gene cloning, molecular cloning, also known as gene cloning. We can use gene cloning in order to isolate specific fragments of DNA. We use molecular cloning to isolate specific fragments of DNA. Remember, this required a plasmid, foreign DNA, and restriction enzymes. I'm not going to go into that much detail for you. Next, we talked about cloning animals. Cloning animals can be done by taking a egg from a donor and mature cells from a recipient. We take the DNA out of the egg. We introduce the DNA from the mature cells of this other donor, or I called it the recipient. We can then grow these into blastocysts and impregnate surrogates for cloning animals. So what do I want you to know? I want you to know that cloning animals is possible. It requires a donor egg, which has been enucleated, and donor DNA from another animal. Great. We also talked about gene therapy. Gene therapy, in order to, it's directed towards curing certain genetic diseases. Gene therapy is for curing certain genetic diseases. And remember, these genes, these fixed versions of genes are delivered by viruses. Delivered by viruses called adenoviruses. Awesome. Lastly, I think we talked about, or second last, we talked about GMOs. GMOs are genetically modified organisms, and these are organisms that can, that have special properties that can allow them to grow in drought conditions, or allow them to grow in the presence of certain pests, etc. And lastly, we learned about gene sequencing. Gene sequencing allows us to determine the genetic sequence of a stretch of DNA so that we know what the gene says, so that we can document the gene sequence and we can solve the genomes of different animals. All right, so those are the things to know from chapter 17, just some very important biotechnology out there, understanding how we can use biotechnology and what are some of the types of biotechnology that are employed. So that about wraps it up. Not a lot of material for the last exam here, but I hope we touched on the important points for chapters 11, 12, and 17. Study this material. I highlighted, I wrote it down here, and that's going to be covered on the exam. I think you can do it. Let's go ahead and finish strong. Make sure you finish all of your homework. I'm also going to post another discussion board, so finish your final discussion board, finish all your homework, finish quizzes and the exams, and we should finish strong. All right, you guys can do it. Until next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.